Scientists in Newcastle are developing cloning techniques that they hope one day will heal currently incurable diseases. Janice has a neurodegenerative disorder. Part of her brain is deteriorating.
I found out in the year 2000 and I decided to have a diagnostic test because my mother has the disorder. It's been in the family for generations. Up until the symptoms started in 2005, I never thought about it even though I'd had the test. But since the symptoms started, I've had to retire from work because it makes one very, very tired and I haven't a lot of strength. And most of the problem is with my speech.
Scientists think that replacing the damaged cells with the new ones will cure Janice's condition. This technique requires special embryonic stem cells. Unfortunately, the easiest way to get them at the moment is from a controversial source: human embryos.
Well, sadly, people have difficulty conceiving a child. We do a procedure called IVF whereby we put the egg and the sperm together in the laboratory, and then 2 days later we can put that embryo directly back into womb to help them achieve a pregnancy. So we give the woman medication to make her grow lots of eggs. So we will often collect maybe 10 eggs from a woman in the process of one IVF, and we put the sperm with them all, and then we put the best 1 or 2 embryos back. So effectively a side effect from IVF treatment is that there'll often be some embryos remaining after treatment. Now they're usually very poor quality, but they're usually then, they would just have to be discarded. We can use some of those embryos potentially for research, and we can create embryonic stem cell lines from those embryos.
Stem cells are originators. They are the beginning of a tissue, and the special thing about them is they retain the capacity to divide constantly and repair themselves. Our body's full of them. We have them in the skin, which constantly replaces our surface covering. The most exciting cells are the ones with embryonic properties. Those are the ones that develop a few days after fertilization of the egg, and that little ball of cells will ultimately form the embryo proper.
Scientists have proved that embryonic stem cells can make the majority of other cell types. Here, stem cells have become heart cells. You can see that they have contractile properties.
Stem cells of embryonic origin are like apprentices that can do anything. They haven't yet been specialized into any single function. And if we can capture them, grow them in a dish, and persuade them to develop under direction, then potentially we could produce any of the human tissues and use that to repair any manner of diseases and injuries.
Scientists believe that in the future, the damage to Janice's brain could be repaired by giving her new cloned brain cells.
The problem is the brain doesn't have its own stem cells and can't repair itself. So the idea is if it's some apprentices, then those apprentices can be placed very precisely into the bit of the brain that's not working, and hopefully will pick up the function of the local cells and start to repair the damage that's been done by the accumulation of iron or other products. Stem cells offer tremendous hope to medicine because it gives us for the first time the chance to explore treating diseases where the tissues have stopped working by producing genetically identical replacement tissue. Something that we've done for a long time in limited areas like bone marrow transplant, but now we can begin to think of doing on a much wider basis.
Stem cell research raises lots of ethical issues, and this makes some people uncomfortable about it, in spite of the potential benefits.
There are 2 major costs to embryonic stem cell research. One is that it involves the destruction often of large numbers of human embryos. Some people believe this is either akin to murder, human beings, or it results in the devaluing of human life. And the second major cost is that we develop technology such as cloning technology that can be used to produce live-born human clones.
I've no problem at all with stem cell research, and I hope one day to be the recipient of it.
This is deoxyribonucleic acid, DNA. It is the most extraordinary molecule on Earth. It literally encodes the genetic instructions from which you are made. It has an amazing base pairing structure which can be replicated, and the information is passed on. Understanding DNA has given scientists new ways of trying to cure diseases that were previously thought to be incurable, like the hereditary respiratory disease, cystic cyst.
I take about 20 different types of drugs, some of which are here, and I also take some of them intravenously, and most of them are done in tablet form. I take about 250 tablets a week, which is 1,000 a month and 12,000 a year. It mainly affects your lungs because the hairs in your lungs don't work properly, so they can't get rid of the mucus in your lungs, and so the tubes get blocked up in your lungs. I have about 30% lung function now. It means that I can't walk very far, I can't really do much, and it affects pretty much everything that I do. Until I was about 12, I was in the school swimming team, I was in the running team, I was in doing PE, I won lots of sports awards. I did pretty much everything that you could do. It is a genetic disease. My mum has one gene mutation and my dad has another one. I just so happened to get both of them at one time, so I got CF. My brother, who's 17, he doesn't have CF at all because he just has two perfectly normal genes.
Gene therapy is when you put in new copies of genes that are defective within patient cells. So, for example, with cystic fibrosis, both the genes have to be abnormal for you to get cystic fibrosis. So what we're trying to do is to introduce a new copy of the gene into the cell.
First scan we're going to do just gives us an image of the whole of your chest, so we can plan where we're going to start and stop the actual scans.
Cystic fibrosis is a possible target because, first of all, you need to know what the gene is, and that was identified in 1989. Secondly, there has to be need for this fancy and probably quite expensive new therapy, and sadly these patients don't have good enough therapy. Finally, you have to be able to introduce the gene somehow, and because we can spray things into the lungs of patients, all three become together to suggest that cystic fibrosis would be a good candidate for gene therapy. The sensible thing would be to do gene therapy as soon as you know you have a diagnosis of cystic fibrosis. And there is now newborn screening happening in the UK, so you could put the two programs together and say, you have been diagnosed with cystic fibrosis, now you need gene therapy to prevent rather than treat. And that's actually what we're very much aiming for. Gene therapy for CF should be a preventative treatment in the future, not trying to stop something that already has deteriorated.
The science of gene therapy provides hope for cystic fibrosis sufferers. In the future, there might be a cure. Tragically, this is too late for Lorna and her family, as she died 2 weeks after giving this interview.